How's everyone doing today? Good? How's the coffee? Excellent. What's the buzz? Thank you. So I'm happy to introduce our next plenary speaker, Dr. Gary Mittelbach. Dr. Mittelbach received his BA from the University of Iowa and his PhD at Michigan State under Dr. Oral Werner. Dr. Mittelbach is now a professor at the Kellogg Biological Station and the Department of Zoology at Michigan State. Dr. Mittelbach has made a tremendous influence in the fields of biogeography and community ecology as one of the most highly cited researchers as declared by the ISI. He is on the board of editors for Global Ecology and Biogeography and is the author of Community Ecology, published by Sinar and Associates last year. His research interests include biogeography, community ecology, the generation and the maintenance of biodiversity, freshwater ecology, and species interactions. So please help me welcome Dr. Gary Mittelbach.
Well, thank you all for inviting me. It's a pleasure to see so many of you here. And it's especially nice to be talking in a symposium run by the grad students. That's always very gratifying. And I hope what I have to tell you today is of interest. So what I want to talk about today, can you all hear me okay back there? Can you hear me if I stand over here still? Yeah, good. I want to talk to you today about patterns of biodiversity and particularly how species diversity, species richness is related to productivity. And I want to talk about both the patterns and the mechanisms. And I want to talk about them at two different spatial scales. One a very large kind of global scale and the other more local. And at the large spatial scale, as he was sitting here somewhere a minute ago, Tom Brooks said a minute ago, we know that biodiversity isn't distributed homogeneously across the globe. In fact, it's concentrated in the humid tropics. Most taxa are much more diverse there. And we've known this for a very long time, since the early, if I can figure out how to run this thing. Yeah, technology. There we go. Since the early explorer naturalists set out in sailing ships a couple hundred years ago, they noticed that tremendous diversity of life in the humid tropics. But and this has become known as the latitudinal diversity gradient. But as Bob Rickliff says in this quote, which is really nice, we still don't know or have a complete understanding of what drives this pattern. Although I hope I'll show you today we know a fair amount more than we did in the late 1980s. Too many buttons. All right, here we go. So we know that diversity is related to climate and that it's related to productivity. All right. If I walk. I'm already over my limit here on technological stuff. So, primary productivity measured here by actual evapotranspiration is much higher in the humid tropics. And for many species of organisms, here for birds, for example, species richness is strongly positively correlated to some measure of productivity. And a few years ago, I was involved, much of what I talk about in the first part of this talk comes from working groups that were at NCEAS, and I was involved in, and Mike Willig was involved in one of these early on. This was one that Brad Hawkins led, and what we did, we looked at the relationship between species richness and measures of climate, usually temperature or water temperature relationships, and find that for these vague groups of organisms, there's a strong predictive relationship, that the single best climate relationship variable explained about 60% of the variance in species richness at broad spatial scale. So climate and productivity and species richness are related at global scales, but what's the mechanism? And so I want to talk about some of those mechanisms today. One might be this mechanism that's been around, hypothesis been around for a while, known either as species energy theory or the more individualist hypothesis. And that idea is that in areas that have higher primary productivity, those areas can support more individuals, and where there are more individuals, there are going to be more species. And it's an idea that dates back to Hutchinson and others. So we can ask, are there more individuals in areas of higher productivity? And when we went to look for this, and this was again part of an NC's working group that Dave Curry was lead on, we actually found surprisingly few data sets where we could look at that relationship on a broad kind of continental scale, but two of them were for trees in gentry plots worldwide, and then for North American breeding bird survey. And in both cases, yes, where there's higher productivity, here measured by AET, there are more individuals for trees and for birds. So that part of the hypothesis is supported. And then we could ask, are there more species where there are more individuals? And again, that's true. Again, for the trees and the birds. So the hypothesis seems to hold, except what we'd really like to know is that how would we expect the number of individuals and the number of species to be related? That is, we see more individuals of these groups as we move to more productive sites. But does that explain the increase in species diversity we see across the latitudinal gradient? And as Alan Hurlbert and co-author noted here in this paper, we don't have theory to guide us here. There's no ecological theory that says for a given number of individuals, we're going to see this many more species. What we have, and what we tried to use, or did use, was empirical relationships. Okay, and if those of you in ecology class have probably heard about this, there's a strong relationship between the number of individuals and the number of species this is known as the species abundance distribution, first studied by Preston and Ronald Fisher. These are distributions that have been developed for lots of different groups of organisms, but they're always kind of at small local to maybe a regional spatial scale. I think the size of Great Britain is about the biggest that's been looked at. But if we use these empirical relationships and apply them at a global scale, which is what we did, we find that, so these are the predicted increases in tree species richness for the increase in number of individual trees across the gentry plots. The blue line is the predicted relationship based upon the Preston log normal. And what you can see is that the predicted increase is far below what we observe. That is, yes, there are more individuals in the humid tropics than in the temperate zone, but that increase in the number of individuals doesn't explain the increase in the number of species we see, at least by this approach. So why? What's going on here? And the explanation, or at least some understanding of that, can be gained by thinking about it this way, okay? In that, for lots of groups of organisms, the more we sample, the more we collect individuals, the more species we find. These are known as collectors' curves, right? And this is a very famous data set. It was first, so they're marine benthic organisms. So Sanders was on ship. They went around the globe. They dropped a dredge over the side of the boat. They pulled up the mud. They counted how many individual benthic invertebrates were there and how many species. And when they did this up near the poles, in boreal waters and in temperate waters, they found kind of a shallow relationship. But when they did the same sampling in tropical waters, a much higher thing. So for the same number of individuals sampled, you find many more species of benthic invertebrates in the tropics than in the boreal zone. Okay? So this is like I said, a famous data set. Bob May reproduced it in his book on species diversity and what did he call that? Stability, species diversity. And then Steve Hubble reproduced it again and looked at it in his book on the neutral theory. So what it says is that there's no one single relationship between the number of individuals at a site and the number of species. Rather, that relationship changes consistently as you move from the poles to the tropics. And another way to think about that, it's a little bit faint, I'm sorry, is to think about it in terms of species area relationships. Okay, again, a very famous relationship. I took this from Rosenzweig's book on species diversity. I think he calls it maybe the oldest relationship in ecology. So we've known for a long time that larger areas support more species. And on the log-log plot, this is generally a linear relationship. Okay? So we can look at the slope of this relationship, and ecologists have spent a lot of time doing that. But what some people have done recently is to summarize the literature and ask, how does the slope of the species area relationship vary with latitude? Does it vary with latitude? And what they find is that as you go, again, from the temperate zone to the equator, that the slope of this relationship gets higher. That is, you find more species for the same area. So that, in my mind, the same thing we saw with the individual species relationship, that there's something about the tropics that allows those habitats to support more species for the same amount of individuals or the same amount of area. So a strictly kind of ecological explanation, the species energy hypothesis, doesn't cut it, okay? It might explain some of the latitudinal gradient, but it doesn't seem to be the whole story. So let's think about evolution, okay? And the ultimate drivers of species diversity, that is, speciation and extinction. And Jablonski wrote a paper in the 50s where he said that any difference between tropical and temperate organisms must be the outcome of evolutionary patterns. Of course, Jablonski would think it would be the outcome of evolution, but I think he certainly was right in that. And evolutionary patterns had been ignored, I think, for a while in thinking about the latitudinal diversity gradient, but not any longer. So here's going to be a quick summary of some evolutionary mechanisms that might drive high diversity in the tropics. One, evolutionary rates might be higher, and here people have proposed that these are rates of base pair substitution, okay, rates of molecular evolution. That is, at higher temperatures or with organisms at shorter generation time, the rates of molecular evolution might be higher in the tropics than the temperate zone. And there's some evidence for this, although the connection between that and speciation rates is still pretty tenuous. Okay. The other possibility is speciation rates are higher in the tropics due to relatively stronger biotic interactions and coevolution. This was Jablonski's hypothesis, and Doug Shemsky's followed up on it. And I'm going to talk more about this in a little bit. And then the idea here is that with work by John Wiens lately and early work by Bob Rickliffs, that many major clades of organisms, trees for example, have tropical origins, and that tropical environments have been around longer in the Earth's geological history than temperate environments. So there's been more time for speciation in the tropics, and if organisms tend to stay, clades tend to stay where they originated. This is the tropical niche conservatism hypothesis, that there are things about an organism niche that prevent it from moving out of the tropics, then that could lead to higher diversity. And then, of course, maybe rates of extinction are lower in the tropics. We don't know much about rates of extinction across latitude. But one thing I do want to remind you of is that we're not talking about, when we talk about the latitudinal diversity gradient, we're not talking about extinctions due to glaciation, okay? One, it's not that all the species in the northern hemisphere were wiped out when the glaciers came down. In fact, most of them didn't disappear at all. They simply moved. There weren't that many extinctions. And the other thing is that the latitudinal diversity gradient is much older in time than the last glaciation. In fact, it's ancient. So paleontologists have done a nice job of documenting this, and I'll just show you some data here from Crane. So these are genera of marine mollusks here currently as we go from the equator to almost to the poles. So many more species of genera of marine mollusks at the equator than the poles currently. And if you look back through a time, and these are paleolatitudes here again, the equator to the poles. So here we're back in the age of the dinosaurs, similar pattern. Here we're back even farther, back to the Devonian, the age of the fishes. Same thing. Okay? So the pattern is ancient. And the other thing is interesting is that it's come and gone many times in the past due to kind of global-wide extinctions. Or here in this case, these again are kind of the darkness indicates the number of genera of marine brachiopods in this case. And here again you see a strong latitudinal gradient that disappeared then in an ice age and then reestablished itself. So when you think about the latitudinal gradient and you think about extinction and time, I just want to plant in your mind that it's been a pervasive feature of the Earth's history for a very long time, which suggests, well, maybe it has a strong general driver behind it, and it's not the result of the extinctions in the last, glaciation. But it is true that tropical environments were much more widespread in the Earth's history and are older. So if we go back to the end, I always get my geological periods thrown. Let's just say we go back 60 million years. I think that's somewhere in the Eocene, when the Earth was very warm. Okay, Texas here, you were basically tropical. Where I live up here, Michigan was subtropical, and the polar oceans were ice-free. So the Earth was very warm at that time, and there's been a big increase in species richness from that time period to today. Now, paleontologists will debate about the slope of this for lots of reasons, but species diversity has increased over that time. So tropical environments are older in the Earth's history and more widespread. And if that's coupled with the idea of tropical niche conservatism, that is that clades or groups of organisms that originated in the tropics tend to stay there, that could be part of the explanation. And I always have trouble with this graph. It looks so simple, but it's a little bit tricky. But this is a recent paper that looked at this idea of tropical niche conservatism and in different major groups of vertebrates. And here, if I remember right, the blue. These are families. Okay. Light blue families are those that originated in North America. And these orange ones are families that originated in South America. And then this is kind of the boundary here between the tropics and outside the tropics. And the point is that families that originated in South America have tended to stay in the tropics, whereas families that originated in North America have both stayed there and also moved into the tropics. So there does seem to be tropical niche conservatism in that the direction of movement through time, dispersal is one way, that things that originated in the North Temperate Zone move into the tropics, but things that originate in the tropics don't tend to come up into the North Temperate Zone. And for probably good reasons. It's hard for lots of organisms to adapt to a freezing environment. So there's certainly support for this idea. So I'd say there's good evidence that time for speciation and niche conservatism contribute to the latitudinal diversity gradient. But I don't think it's the whole story, and I get into some discussions with people about this, and I don't think it's the whole story for a couple reasons. One, if that's true, then wouldn't we expect that, organisms and groups of organisms should, on average, be older in the tropics than in the temperate zone, if they've been there longer? But in fact, in a nice study here published by Jablonski and all, they titled it Out of the Tropics, they show that genera of marine mollusks, if we look at their mean age based on fossil evidence, that here in the tropics, the mean age of genera of marine mollusks is actually younger than in the temperate zone. And in fact, what Jablonski et al. Found, and why they call their paper Out of the Tropics, is that for marine organisms, a lot of origination of taxa occurred in the tropics, and then they migrated out into the temperate zone. So they view the tropics as kind of a center of origin and then movement out, which is exactly opposite of the niche conservatism idea. So I've covered a couple explanations for the higher richness in the tropics and the temperate zone, the species energy hypothesis, the time for speciation hypothesis, and what I want to talk about last here is the idea is that do rates of diversification, speciation minus extinction, are those higher in the tropics than in the temperate zone? And if so, even if they're of the same age, those habitats, we'd expect to find more species in the tropics. So what's the evidence for that? Well, there are two places we can look. We can look at fossils and we can look at molecular phylogenies. And the fossil evidence is pretty clear, I think. So here, that showing that rates of diversification tend to be higher in tropical habitats than temperate habitats. So here are data for marine foraminifera. So these are tiny little protozoans that form these hard shells that have these really interesting shapes and make wonderful fossils. So a whole lot of things. Climate data, past climate data, is a lot of it's based on the shells of these foraminifera. And here, again, we can look at in the tropics and in the temperate zone over the same time period, rates of diversification of foraminifera were much higher in the tropics. Oops, what happened to my mollusks here? They disappeared. Anyway, these are shells, bivalve shells anyway. But again, some data here, again showing that after this mass extinction, that rates of diversification, increases in richness, were much higher in the warm waters of the West Pacific than in the Arctic. So the fossil evidence is pretty clear, I think, showing higher rates of diversification often in tropical habitats. The other way we can look at it is with molecular phylogenies, okay? Because molecular phylogenies that are time dated give us estimates of rates of diversification. Although there's lots of pitfalls here. I don't do this work personally, so don't expect me to know anything truly about this. But I can tell you what people have said. And here's a great study that appeared just last year in Ecology Letters by Condamine et al. And they looked at rates of diversification of swallowtail butterflies. And these guys are really cool. Okay, so in the New World, the Western Old World, and the Eastern Old World, they show a really strong latitudinal diversity gradient, highest diversity here at the equator. And what was really cool about these guys is that there are two major clades I think these were in the New World. They originated here in the Eocene when I told you that the world was very warm and wet and tropical environments extended up into this area. In fact, the evidence is that they originated here, both clades. One clade stayed temperate, whereas the other migrated down into the tropics. And then both clades diversified through time. And what you can see here in this lineage through time plot, so this is increase in diversity over time from the Eocene to the present. And just to point out, this is a proxy of temperature here, so it was warm and then it's gotten cooler. So we can see that the temperate clade diversified through time to about not quite 80 species. But over that same time period, the tropical clade diversified to about 120 species. So nice data from molecular phylogeny showing that rates of diversification of these two sister clades that originated at the same time. The one that was tropical diversified much faster. And of course, we don't know whether that's speciation or extinction or some combination of both. All right, I'm not going to go through all of these. You don't have to be scared about that. So these are what are some possible mechanisms for why rates of diversification should be higher in the tropics and in the temperate zone? And there are lots of them, and they deal both with speciation and with extinction. Okay, and I'm just going to focus on one. Okay, and not because it's the best, but it's maybe one you haven't heard of, and because Doug Shemsky is a friend of mine and colleague, and I've worked with him on this. Okay, so this is the idea that Dobzhansky proposed long ago, that stronger biotic interactions in the tropics might promote greater rates of speciation. And what Dobzhansky said, that these stronger biotic interactions in the tropics increase the opportunity for the evolution of novel forms. And how Doug has expanded it is to say, and I'm not very good at explaining this, but the idea is that, in the tropics, the environment is mild and amenable to life in general, and that therefore most of the selective force acting on organisms is not adaptation to the abiotic environment, but adaptation to the interactions with other species, the biotic environment. And if that's the case, then because the abiotic environment is kind of a fixed target, in a sense, compared to the biotic environment, that species are interacting with other species, and that might be at different points on their range, or that other species that co-evolve through time, and therefore adaptation becomes a moving target. The argument is that coevolution and then this biotic drift could lead to greater selection pressures on species, especially at different parts of their range, to diverge in their adaptations and potentially to speciate. So that's a short explanation of a somewhat complicated idea. Oh, I forgot to mention, I fish with Doug a lot, so that's a nice smallmouth from Lake Erie, and he'd be happy if I showed his picture with a big fish. Anyway, so we did a review article a few years ago, not to test that hypothesis, because that's something still out on the horizon, but just to ask this more simple question, the more basic question, is there a difference in the strength of biotic interactions or the importance of biotic interactions as we go from the temperate zone to the tropics? And what we did in this review was to survey the literature of studies that had looked at species across some latitudinal gradient, at least 10 degrees, and most of them span that. And then we had about 90 studies in our review. And then we try and look at the strength of biotic interactions. This turned out to be really a diverse, heterogeneous literature that was kind of a mess. Kind of still is. But so the best we could do was to group interactions into these four kind of broad categories: those that deal with predation and parasitism, disease; those that dealt with defenses against predators; those that dealt with mutualisms; and then other indications of biotic interactions. And I want to note that surprisingly, there are almost no studies out there that look at the strength of interspecific competition across a latitudinal gradient. That's just remarkable, Not that it's easy to do. It isn't easy to do. But some species interactions are very poorly represented in the literature across latitudinal gradients. And then the other caveat I want to make is that it's hard to separate cause and effect. If we find stronger biotic interactions in the tropics and the temperate zone, is that the cause of greater diversity, or is it the outcome of greater diversity? Okay, and that's a point made by Fisher quite a while ago, and I'm not going to satisfactorily address that today. All right, so what do we find? Well, if we look at just kind of the frequency of occurrence of interactions, and just go through some examples here of mutualisms. One, things like cleaning symbioses are much more common in tropical systems than in temperate systems, and they're well developed in the tropics. So that on tropical coral reefs, you know this probably, there are these cleaning stations that fish come to visit. Big predatory fish will sit there and let themselves be cleaned by smaller fish that could become their meal, but instead they don't. There's a nice strong mutualism there. Pollination, okay? The frequency of animal pollinated plants, much higher in the tropics than in the temperate region. Seed dispersal, another biotic interaction. 90% of tropical plant species are animal dispersed, and that is a much higher percentage than in the temperate zone. And then plant-animal mutualisms. These are tight symbiotic mutualisms. They're almost absent in the temperate zone, and yet in the tropics, they're really common and have co-evolved, have evolved in many different groups. Over 1,000 plant genera, over 100 plant genera, and over 40 ant genera. So those are just some patterns. And I just want to briefly show this current study. I'd like to think that our paper had some effect, and maybe is having some effect, on people looking for these types of patterns. And this is a nice paper that was just published a little bit ago by Salazar and Marquez, who looked at herbivory in these two species of tropical shrubs and small trees. And again, the number of herbivore species and the abundance of herbivores per area of plant. They found that it peaked at the equator, actually, and dropped off on both sides. Okay, so there's more herbivore pressure at equator and drops off on the sides. So we've got these comparative studies, but what would be really cool is if we had more experiments, okay? Just to do an experiment across a broad latitudinal gradient that said, let's set up the same experiment across this gradient and ask, how strong is predation across that gradient, or how strong is competition, and measure that. And here's a wonderful example from a long, long time ago of somebody who did that. Okay, so Jane, he used these the larvae of Polistes wasps here, and they're predated upon by ants. So he had this common prey item, and then he set out in a really pretty big experiment, wasp larvae in a number of habitats and a number of sites from New Hampshire down to Brazil. So a broad latitudinal gradient. And then asked how long it took ants to find and eat the wasp larvae. And what he found was that the time of survival was much lower in tropical habitats, Brazil, here, Costa Rica, than in New Hampshire. And he concluded that predation by ants becomes greater towards the tropics. And so a simple experiment, and how surprising we have so few of these. But there are some more recent ones, and I point you to some nice papers studied by Amy Freestone and colleagues looking at intensity of predation in marine intertidal systems. And Bruce Mengi and Jane Lopchenko did some early work on this long ago, comparing Panama to North Temperate intertidal. But we need more of these studies. So what's needed? Many more controlled experiments. Do them in a way where we can measure effect sizes and do a meta-analysis. Obviously our review is not a formal meta-analysis. We need more comparative studies still. We can look at traits that are correlated with biotic interactions and look at their patterns across latitude. And then distinguish cause and effect. Be nice to figure out what drives what. And then just a quick quote, because this has taken a while. Despite the enormous literature on biotic interactions, Jablonski says, we still know very little about them as agents of macroevolution. Again, surprising. And then the other thing is that even if rates of speciation and diversification are higher in the tropics than in the temperate zone, we still wrestle with this question, well, how do all of these species now coexist? We still have to get around that question. How do they coexist in the tropics? But I think here a nice quote from Vermeer is basically the idea that species become niches for other species. And maybe it's not such a hard thing after all. And I'll point you to this interaction in the tropics. These are fire ants, okay? They go marauding through the, army ants. They go marauding through the forest floor. They stir up all kinds of insects when they do that. This is a group of birds that follow the army ants, feed on those insects, and then there's a whole group of butterflies that feed on the droppings of the birds that follow the army ants. So again, species become niches for other species. So maybe that tropical diversity and the coexistence isn't so hard to explain. How much time do I have? I've got time. 23 minutes. We'll move along. So I want to leave the broad scale pattern of productivity and diversity and move to more local, regional scales, finer scales. So at the broad scale, here's what I've tried to say. Some measure of productivity, potential evapotranspiration here is related to species richness. I think this was plant species richness in this study. And it's positive, although it's often decelerating, but it tends to be positive. At local scales, though, that relationship is much more variable. And I'm sorry this is a little bit fuzzy, but long ago, we did a review of this literature, and Mike Willig was involved with me in doing this study at NC. So what we thought, well, as a bright idea, we had all of these LTR sites that measure productivity and diversity of different taxa. We thought, well, get all these data together, we'll analyze them. And at the time, productivity and species diversity was thought to be a hump-shaped relationship. Dave Tillman said that, Mike Rosenzweig said that emphatically. So that's what we figured we'd find. And to our surprise, we didn't. We found, so looking at kind of different spatial scales, local to landscape and then broader regional and then big continental to global, we characterized the relationships we saw, this is for plants, we also did it for animals too, as either positive, like this, or negative, or humped, meaning it goes like this, or some U-shaped ones, which we were surprised to find, or non-significant. And what we found was that at big spatial scales, things tend to be positive or humped. And when I say humped, they weren't like they went like this, they often look like this. So depending on how you did the statistical analysis, this was either monotonic or humped, and that led to a lot of controversy. But at smaller scales, we found a whole variety of relationships, and humped were more common. So we sent this thing off to ecology, got rejected. We sent it back to ecology, it got rejected. We sent it back to ecology, it got rejected. I think, Mike, we submitted it 4 times. 6 different reviewers. So we had a lot of trouble publishing this study. We finally persevered. And the reason we had trouble publishing it, I think, is that we didn't find enough humps. So we published it, and then immediately we got criticized, and people reanalyzed the data to say we were too generous in looking for humps. So Gilman and Wright applied stricter criteria to the studies they put in there and to different statistical analysis, and they found more positive relationships than we did. They basically are looking at the same data set, and fewer humps. And then, this has been quite a saga, there was a forum in ecology a couple years ago that kicked off by Robert Whitaker, who said, I'm tired of these guys analyzing these data sets. None of them are worth a darn. We should just call time out on any more meta-analysis of productivity and diversity. And basically said that data were just too heterogeneous to really make sense out of. And then there was a whole series of responses. I did a short response, and a lot of others talked about meta-analysis and whether it was worthwhile. And the response was basically, well, meta-analysis is a good thing, even though the data are sometimes heterogeneous. It's still our best tool. And I don't know what people think about the productivity diversity pattern from that. Lots of it. So anyway, before I jump to that, okay, so that's kind of the background, that at local scales, small scales, we get all kinds of different relationships, unlike what we see at very broad scales like the latitudinal gradient. So there was a study done just a few years ago published by Peter Adler and Eric Seelblum and I can't remember some of the other people. They have this nice set of experimental sites scattered across the globe, and these are small plots to look at plant diversity and productivity, small 1 meter plots at 48 sites worldwide. And it's kind of a grassroots thing that these guys have done. But they analyzed their data, here measuring productivity as live plant biomass, and then species richness, and then looked at the pattern at each of those sites. And that's what all of these different lines are. So they found negative relationships, positive relationships, a couple humps, some U's, and lots of non-significant relationships. So their point was that, okay, now we don't have a heterogeneous data set of lots of different methods and lots of different types of analysis. We did the same experiment, same observational study everywhere across the globe, and we find that productivity is a poor predictor of plant species richness. That's their conclusion. And they've been commented on, too, on this. And while it is true, I think you might want to say, well, if we just look at maximum species richness across productivity, well, there does seem to be something there, okay? So and that's worldwide. So what's going on here at each local site is a little bit hard to figure, but there may be some bigger pattern. I leave you with all that unsatisfyingly, just to say that I think at small spatial scales that the pattern between productivity and species diversity is varied, maybe because there are lots of different mechanisms that may be driving it, probably because of that. But what I want to do in the little bit of time I have remaining, and it's not much, is to talk about this, that we know that this is a very strong pattern. If you add nutrients to plant communities, terrestrial or aquatic, species richness goes down. And we know this from the longest running experiment in ecology, the parkgrass experiment at Rothamsted. When you add fertilizer to plots, species richness drops over, I think the study has been going 150 years or something. And it's not an idle, or this is my connection to theory and practice. Isn't that what we're doing? This is what they're thinking. This is my little connection to practice, in that nitrogen deposition, nutrient deposition from the atmosphere is a big problem, especially in the northern hemisphere, and that with nitrogen deposition and increase in fertility, species richness in many plant communities has been declining. So that pattern is very strong. But again, what's the mechanism that might be driving it? I'm going to have to cruise right along here on this and say, well, one possible mechanism, we can think about it in terms of kind of a Tillman-esque way of thinking about it, is that if spatial heterogeneity in nutrient patches, high and low nutrient patches across a field, for example, if that allows different species of plants to coexist in terms of competitive interactions, then if deposition of nutrients evenly across that field, either through atmospheric deposition or fertilization experiments, might reduce that heterogeneity and decrease diversity. And so we set out to test that hypothesis. My co-PIs, Kay Gross and Heather Reynolds, we did an experiment, 2 experiments here, located in a native sandplain plant community near Lake Michigan, where I work here at KBS is right here. We set up an experiment where we manipulated productivity by adding fertilizer, and we did it either uniformly or in a patchy way. So we took 3 by 3 meter plots, divided into 144 of these little grids, that are 12 by 12 centimeters, and then applied fertilizer in a patchy way at the same overall level as the uniform way. And our hypothesis was that when we applied fertilizer uniformly, diversity would go down, but when we applied it in a heterogeneous way, patchy way, diversity would be maintained. And so the effects of adding fertilizer were obvious right away. You know, here we set it up in April. Here's in June. You can see these high fertility patches have more biomass. And diversity went down over 10 years. So this is plant species richness, control versus fertilized. But notice it went down the same in the patchy and in the uniform treatments. So heterogeneity didn't seem to have an effect. But what we did find was that not all plant species responded the same way. That clonal plants, those that reproduced with rhizomes and stolons that spread kind of like gorilla-like across the field, those guys did really well when we fertilized compared to non-clonal species. And it was the non-clonal species that decreased in richness with fertilization and not the clonals. So that suggests that there's something about maybe these plant traits of clonality that's involved in the mechanism of species loss. So these are some of the clonal species that are common in our area. This is quackgrass, and this is like a bayberry. And again, just to make the point that where there's more clonal biomass, species richness goes down. So we set up an experiment where we specifically manipulated whether clonals were present or absent in the community. So we had two types, with clonals and without, and then different fertilization. So we had the same uniform small patch and then a large patch fertilization treatment. So we set up this community. This time we rounded up the whole large area of the field to start the communities from scratch. We didn't want any kind of historical effects here. So we did that for about a year, and then I didn't do much of this at all. Okay, you won't see me in many of these pictures, but so that we started the communities from scratch with seeds of about 40 native species of non-clonals, and then I think 8 species of clonals in the clonal treatment. Here's the field crew. That's Kay, that's Heather. I could say that's me, but I don't think so. Anyway, so what we found was that, okay, I'm going to separate the non-clonal community as those 40 species of native plants that don't have clonality, and then the mixed are those same 40 species plus 8 species of clonals. Okay? And that in unfertilized treatments, here the control, species richness goes up through time. Okay, the communities are being colonized and developing. And that with fertilization, so these are the large patch fertilized, small patch, and uniform, we can see that in the absence of clonality, increasing nutrient richness, decreased species diversity, okay? Not very much in the large patch, more in the other treatments. But in the presence of clonals, that difference was much greater, okay? When clonals were present, diversity species went down in the large patch some and actually decreased through time in the uniform and small patch treatments. And so there's actually an interaction amongst all the treatments. 10 minutes left. We'll make it. We're going to make it here. So species richness, total species richness in the plot, when we, in an unfertilized treatment, when we added clonals, species richness was higher. Okay? But when we fertilized that same community, species richness actually decreased more. So in the presence of clonals, we actually got less species than in the absence. And that effect, that loss of non-clonal diversity was greater when we fertilized in small patches and large patches. So what's the mechanism? Well, I think it's competition for light. So what it looks like is that when we add fertilizer, okay, the amount of light reaching the base of the canopy declines. When we add more fertilizer, there's basically more plant biomass. It declines more in communities with clonals than without. And it's interesting, so species loss is directly related to the amount of light reaching the base of the canopy there, that where there's less light, we get less species, and that communities with clonals and without fall along the same line here. So clonals drive down light to lower levels. They make more biomass, they drive down light to lower levels. But interestingly, in communities with clonals, they leave behind more nitrogen. Okay, so if we look at the amount of nitrogen available in the soil, it's actually higher in those clonal communities. So there looks to be a trade-off here, that clonals are really good at depressing light, okay, and depressing species richness because of that, but they're poor at utilizing perhaps soil nutrients. So I think what's going on, what we think is going on, is that because clonals can provision their offspring with resources from the parent plant, they put out these stolons and rhizomes, that they're better able to recruit in a low light environment than are the non-clonal species. And that's maybe what's going on, although we don't have evidence for that. And how general is this? Well, you know, maybe this isn't the mechanism of species loss in all plant communities with fertilization, but there is some nice evidence coming out that, you know, when you fertilize, these are the fertilized treatments, that the abundance of these tall runner species or tall clonal species increases in communities. And this is across all the LTR sites. So I'm zooming along here. Okay, now we're at the end. General conclusions. Species richness is correlated with productivity, but the pattern differs across spatial scales. At continental scales, species richness is positively correlated with climatic variables that drive productivity, water and temperature. And there are multiple mechanisms that are responsible for the latitudinal gradient and why the response to productivity. I think these are perhaps the main ones: time for speciation, niche conservatism, and probably, I would say, higher rates of diversification in the tropics. All of these need to be tested more. And at small spatial scales, the point is that that productivity-diversity relationship is much more variable. I think there are many more mechanisms that can be involved, but that if we add nutrients, eutrophication, it almost invariably leads to species loss. Well, almost invariably. And again, mechanisms, we've tried to look at some in plant communities, and there are no doubt others. I thank you. If you want to read any more about some of this stuff, I put in a shameless plug here to buy my book and read my book. But mostly I just want to say thank you. Do we have any time for questions, Warren?
We do have time for questions. I have one.
Yep.
What comments? I really enjoyed your talk, thanks. What comments do you have about the relationship between alpha diversity and beta diversity and the role of beta diversity in influencing species richness at different scales?
That's a really good question. And I don't know if I have too many comments about that. I mean, people have certainly looked at that, you know, kind of across things like the latitudinal gradient to address whether, you know, the response is there's difference in beta diversity across there. And I can't pull that up right now. Do you remember how that goes in general?
I just know one study that we did showed that perfect form diversity in Costa Rica was really largely explained by these complicated patterns of beta diversity among sites that ultimately is related to the configuration of landscapes at multiple scales. So you have variable landscapes in a smaller area, and that leads to more beta diversity. And I don't have any mechanistic cause to explain that, but when the patterns of beta diversity are not strictly correlated with distance and things like that, it seems to go a long way towards explaining why there are so many species at the scale where you're looking.
Right. Yeah, you're absolutely right. And just looking at diversity itself, and then instead trying to partition those components of diversity, alpha and beta diversity, is an important next step. And there's been also work done on how productivity might affect beta diversity at local scales, the kind of Chase and Leibold kind of work on that, where at higher productivity sites there seemed to be, again, more beta diversity. Yeah, it's a real good question. There was another one in the back a second ago. Yeah, or in the mid-back.
My question is actually sort of similar, but on the log series scales going across larger ranges than, say, the size of England, you would start to run into areas that sort of have different deep evolutionary history for clade divergence.
Okay.
And that, if you had some way of knowing when you crossed those boundaries, you could maybe correct for that and see what the effect of widely divergent evolutionary history is relative to just area on species binding. So just something like that to...
So you're saying as we move across these broader geographical gradients, you're moving from, kind of one clade to the next clade, or different groups of kind of pretty unrelated species across there, and that it might affect these individual diversity relationships. Good point. And so you think maybe there's some way to try and deal with that or correct for that?
I think if there was some way to correct for it, it might inform a little bit about what some of the actual underlying mechanisms for the latitudinal diversity gradient might be and how much they're related to evolutionary history versus... I'm not quite sure where you go from there, but...
Yeah. No, I think that's a real good point. Absolutely. Anything else?
I also enjoyed your presentation very much.
Thank you.
In your survey of the literature on tropical biodiversity, any empirical or theoretical evidence about the categories or groupings of biotic interactions that may be more important in promoting speciation?
Oh, that may be more important in promoting speciation.
Yeah, you listed
Right.
them broadly, and I understand they're not
Right.
documented that well, but any evidence of particular groups of interactions?
I mean, certainly the feeling, and probably correctly so, although not as well documented, is that mutualistic interactions are more prevalent, more important in tropical environments than in temperate environments. So positive interactions. And to the extent, I think certainly Doug Shemsky feels that those are the ones more likely to generate kind of this coevolutionary feedback and potentially speciation driven. The mutualisms rather than the negative interactions, the competition and predation. But yeah. Do I still have time?
Quick question.
Okay.
So you sort of rule out the effect of glaciations, or maybe it's not a very strong effect on the pattern. But I was thinking, couldn't there be some synergy with, for example, you talk about the tropical niche conservatism.
Yes.
What if you have species that are sort of tropical, subtropical, whose ranges when it's warmer are they reach higher latitudes, but when you do get glaciations, they shift, but they also their populations almost crash. Their effective populations sizes are lower, and their rates of diversification due to that are also lower.
Okay. Yeah, there's lots of possible dynamics going on. One that's been proposed is that those species that are temperate, because they witness these changes in climates, glaciation and stuff across time, are kind of more broad in their niche because they're more used to dealing with these climatic fluctuations, and therefore, in a sense, might be less likely to speciate because they're more generalized, less specialized. If I'm getting that argument right, I think that was it. But yeah, so I think while glaciations, they're just thinking simplistically about their direct effect as agents of extinction probably isn't too worthwhile. Thinking about them in terms of how the species that interact with that and their niches develop is an important point. I think I got to get both.
Let's not